So after our very nice plenary lecture, we're going to start the conference with a workshop, and it is my pleasure to be the chair of this symposium on the new advances in the evolutionary neuroscience. The first speaker is Katherine Bryant. She's coming from the University Aix-Marseille, and she's an expert on neuroanatomy of non-human primates, brain neuron, on the brain anatomy of non-human primates, and she's going to give us some insights of this in relation to the evolution of language. Thank you very much.
Thank you very much. Thanks.
Okay. Sorry, before we start, yes. I just want to say to the people that are online that we are looking forward to your questions. So I will be here looking at the chat. So if you want to ask questions, please just write them down there, and I will be asking the question for you. Thank you. Yeah. So the floor is yours. Thanks.
Thank you. Thanks for inviting me. I'm excited to be here. So as Ruth said, I'm an evolutionary neuroanatomist. To give you a little background on me, I started out as a biologist and then an evolutionary biologist, and then got very interested in human and primate similarities and differences, and have sort of combined the two areas to now look at neuroscience and neuroanatomy, but with an evolutionary lens. So I thought today might be a good opportunity to talk to you all about something that is of special interest to me, which is the role of the temporal lobe in human evolution and in primate evolution more broadly, and how understanding certain brain structures from an evolutionary perspective might be useful, not only for understanding our place in sort of the biological world, but also for understanding human cognition, behavior, and perhaps even helping us understand human brain disease. So I'll be talking about the temporal lobe as a locus of evolutionary change in primates. So first I'm going to talk about what I mean by the primate temporal lobe. Then I'll talk about applying comparative anatomical approaches to understand brain organization, which is something I'm sure other people in our session today will also be discussing. So I'll tell you a little bit about my approaches. Then I'll zoom in a little more into the human question and talk about what this means for the human temporal lobe, what specializations I think that we have in this structure, and then I'll have some ideas for future directions for where I think this research could take us. So essentially, the primate temporal lobe evolved once. It evolved very early. It evolved sort of at the debut of primates as a mammalian order. So this is a fossil primate here. It's a member of the Homomyidae. They existed something about 55 million years ago. This is when they emerged, and they would have emerged at a time when they were splitting off from several other mammals that had sort of similar lifestyles. So there's something called a colugo or a flying lemur, which you might be aware of, that is also an arboreal mammal that these guys were closely related to, but they started existing in a different niche. So we know a little bit about them. We know that they lived in trees. We are pretty sure that they were specializing in a niche that involved foraging in sort of the terminal branches of angiosperms. So at the time, trees were evolving that had flowers and fruits, and these animals, we believe, were evolving to exploit these resources that were only at these very edges of branches. So they're extremely arboreal, involved in some pretty complex spatial navigation. And they also, we think they were exploiting and eating fruits and flowers, although it could have also been insects. But whatever it was, that's where they were located. But obviously, we don't have brains from these animals, but we do have what are called endocasts, which is essentially if you have a high-quality fossil skull from an animal, you can essentially make a mold, pull out the mold, and the way the decay and the fossilization processes happen, there is still an imprint of the brain that had been in this skull. So it's not perfect, but it actually gives us more information than you might think. So here's a reconstruction of what the brain of these fossil primates would have looked like based on these types of imprints. And already we see the beginning of what is called here Sylvian sulcus. We might also call it the lateral fissure or the sylvian fissure, and this is a hallmark of primate brains. So it evolved extremely early, and it's what delineates the temporal lobe. So some of my colleagues and I have argued that this is what makes a primate brain. A primate brain is the delineation of this part of the brain separately from the rest of the brain through this sylvian sulcus. We can also look at living primates to see evidence for whether or not the temporal lobe is a primate unique structure. I want to add a little caveat here. If there's other people who study other mammals besides primates, we do call things temporal lobes in other primates like carnivores, and I'm not trying to say they aren't temporal lobes, but I am arguing that they emerged in a different evolutionary lineage and they have a different organization. So although on a gross morphological level, a lateral expansion of cortex that's associated with auditory cortex is one definition of a temporal lobe, I'm arguing the primate temporal lobe is something that emerged a single time in primate evolution and is possibly organizationally different than these other animals' temporal lobes. So if we look at extant primates, if we want to use maximum parsimony, we can also argue that, well, it appears that every primate across this lineage has something that has a sylvian fissure and has a delineated temporal lobe. And you might even notice that they're pretty large, actually, in comparison to the rest of the brain, especially in these smaller species. So this is just to make it look a little easier for you to see. So in these prosimians and other kinds of old world and new world monkeys, a temporal lobe is always visible. Whereas in the Dimoptera, which is our colugo or our flying lemur, we don't have a temporal lobe. We have a different structure up here. I wish I knew more about it, but I don't think anybody's done a lot of work on those guys yet. So if you have one of those brains, let me know. Another interesting thing, which I'll talk about in a second here, is once we look at the temporal lobe, we also see there is some variation in its lateral sulcal morphology. And one interesting thing is we often see this STS or superior temporal sulcus emerge early in sort of like the process of, as brains expand, if there's sort of an encephalization process happening, a larger animal is going to have a larger brain. And as that happens, we tend to see more sulci. It so happens in primates, those sulci usually start with an STS or superior temporal sulcus. And then as you go into animals that have been selected to have larger and larger brains, you see more and more sulci. So you get much more complex looking temporal lobes in humans and our great ape and lesser ape relatives. So what's going on in the temporal lobe? Well, I think most of us know that there's auditory information in here, and there's going to be auditory association cortex around here. But below the STS, there's a lot of visual processing happening, not just in humans, but in other primates as well. So this has been really, well studied in macaque monkeys, understanding how visual processing occurs starting from primary visual cortex and as it moves along the temporal lobe. So I'm sure many of you are aware of the idea of the dorsal and ventral visual streams, but this has been one very interesting feature of understanding primate brains is the way that as visual information is processed and moved forward in the chain, it kind of more or less splits into two different streams. So we have a dorsal stream, which is very concerned with larger objects. It's concerned with less fine-grained information, and it's very concerned with movement and spatial location of objects. So one might argue it's very good at seeing large objects that are moving, and it's very sensitized towards that. The other stream is the ventral stream, and this stream is getting information with a bias towards cones and other fine-grained information coming from the retina. And what this means is it's really biased towards processing objects. So it's good at processing texture, it's good at processing color, and it's involved in essentially object recognition. So that's what's going on in macaque monkeys. It's also what's going on in humans, and we have the same system as they do, again, with this sort of ventral stream of visual information that travels along the temporal lobe. So this is getting better, but years ago, we really only had, if we wanted to compare, for example, temporal lobes or any other brain structure in humans, we would compare them to macaques because that was the most used model for understanding brains or primate brains. And already there's some pretty big differences. For example, humans have an additional major sulcus going along the inferior part of the temporal lobe, which macaques and many other old world monkeys do not. So this is kind of a problem because what it means is we can't infer the differences that we find between humans or macaques. We can't determine whether it's a macaque specialization or a human specialization. We don't have an outgroup. All we know is that there's a difference. we don't know what the ancestral state is. So one of the points of evolutionary neuroscience is it basically creating a series of outgroups. And as you pull in data from different outgroups, you can start to determine using parsimony what's the most likely evolutionary trajectory. So for example, let's say we were completely naive about temporal lobes and we see, okay, humans have these two sulci, macaques have one. Well, we don't know which came first, right? Maybe the macaques lost theirs, or maybe we gained ours. Well, now we see, okay, if we look at chimpanzees, they also have a similar sulcal orientation as we do. So we can start to go back and say, oh, it looks like humans and chimps had a common ancestor that probably had this, whereas old world monkeys don't. We look at other monkeys, we see generally they only have the one. Then we can start to make some basic logical understandings, but you might say, well, that's not terribly interesting. So you found an extra crease, who cares? Well, we can do way more than that. So one of the exciting things about just starting from gross morphology is at least it gives you a landmark where you can start to say, well, what's going on in this gyrus, or what's going on in this sulcus? What's new, or what's just simply a brain expansion and it's not a change. So already with comparing chimps, humans, and macaques, we can see some interesting differences in temporal lobe organization that are beyond just these sulcal differences. So this map here with the hot to cool colors is a myelin map. So there's a type of neuroimaging where you can get myelin cortex information. The myelin content of cortex. So you'll see, for example, primary areas like motor areas, visual areas, auditory areas are hot red. So primary areas are going to be more myelinated. As you get more and more into multimodal processing areas, association cortex, it's less and less myelinated, so you get these cooler colors. So already we can see there's a big difference in the amount of the relative myelin content in these cortex compared to macaques, where there seems to be an expansion of lightly myelinated cortex in great apes, which to me suggests this is an expansion of association cortex. So we're taking primary sensory information and we're doing more perhaps unimodal and perhaps multimodal processing sort of downstream. The other thing we can see with this additional sulcus is we have this middle temporal gyrus. This is really interesting because in humans, this area essentially houses partially Wernicke's area on the left-hand side, so obviously a very important language area. So that tells me, well, as this temporal lobe expanded and became more folded, it started adopting new functions. But the nice thing about looking at chimps is we can say, well, they have an MTG too. What is it doing? We can't put them in the scanner, unfortunately, but we could do other kinds of examinations of organization to see if there's similarities and differences. So in terms of the entire temporal lobe, it's larger in great apes. We have the MTG. We also have this development of this gyrus on the ventral surface of the temporal lobe called the fusiform gyrus, which in humans houses the fusiform face area, visual word form areas, a lot of really discrete visual processing information or visual object processing. So the fact that chimpanzees have a similar shaped gyrus in a similar location suggests that there may be some functional implications for this. And finally, the anterior temporal lobe, which we don't even call this part of macaque brain's anterior temporal lobe. I don't know whether or not I agree with it, but I think it's partially to recognize that it is expanded. It's larger in humans. We see something similar in chimpanzees. Because we know the anterior temporal lobe is so important for human semantic processing and social cognition, that also gives us some ideas about what might be unique about human brains, how they're different from macaques, and what we may or may not share with chimpanzees. This is just an example of how you can use phylogenetic approaches to then integrate information about function or behavior. So here, this person is not doing any neuroscience really, they're not doing any neuroanatomy, but they're doing behavioral research or cognitive research looking at the differences in behavior across primate species. So another thing we can discover if we bring in enough species is we can find also parallel or convergent evolution. So here we find some interesting complex social behavior in terms of recognizing kin versus non-kin and whether you want to cooperate with them. And we see that, well, we understand that great apes seem to evolve that. We all seem to have it. So the most parsimonious explanation is that it emerged in one of our ancestors after we split from orangutans, but we also see it in cebus monkeys. So that tells us that this is something that can emerge uniquely in another group or convergently in another group. So, okay, how do we do some anatomical approaches? Say we have behavioral data, we have cognitive data, or we have a hypothesis about what might be different between species, but we want to compare their brains. So what are some ways we can do this? So the types of data that I work on are diffusion-weighted imaging. So if you're not familiar, this is a form of MRI that captures information about the movement of water in the brain. And so what that gives us is a proxy for understanding connectivity, because water is hydrophobic, it moves along myelin. It gives us information about the organization of myelin in the cortex, and it gives us direction. So the colors indicate which direction it's moving in. So this is just an image of what the scans look like when they're processed. And this is a very nice tree that has been created by folks at University of Oxford, who are in the process of collecting and scanning post-mortem brains from a wide variety of primates that are not well studied, and basically building out a tree to start the process of understanding brain evolution in primates. And by the way, if you're interested in this kind of work, it's available. You can request access to these kinds of data. So they really want to collaborate with people who are interested in this. And these are analyzed with different kinds of tools. I use FSL, and which is a package at University of Oxford for processing these kinds of data. And essentially, what we've been doing is building ways to reconstruct major white matter pathways. So the major white matter pathways, like fasciculi, we know where they are in humans very well from blunt dissection work, and we know very well where they are in macaques from track tracing work. So we're able to extrapolate from that information and then start saying, well, what would the tracks look like in all kinds of primates? We know the basic tracks. So we build what are called protocols. And so what I do here is I use an anatomically informed approach. So I use sulcal landmarks and other brain landmarks and as well as data from the imaging, the visual parts of the imaging, to see where the core of the tract is, and I develop a way to reconstruct the tract. So essentially I say, I want you to start here and end here, I want you to avoid these areas, and I'm able to reconstruct major tracts from a whole postmortem brain without doing dissection. And we also we create these protocols, and as we create them for each species, we add them to a program within FSL called Extract, so people with brains in this species can use these protocols as well. And so the goal of doing this kind of what we can call virtual dissection is to create a white matter atlas or a white matter map. So start to say, okay, what if we can do maps for any kind of species? We have a common set of rules of how we're going to reconstruct the fasciculi, and then we can directly compare them. So we create a map for our approximately 40 tracks in one species. We provide the recipes to create them, and then we can create essentially a series of maps that people can use to understand the organization of the brain across species. But we can do more than that. So say okay, well, that's fine, that's volume. Maybe I'm going to look at volumes of tracks or where the tracks are going, but I want to know more about the relationship of the tract to the cortical surface, because say you're interested in a certain cortical area, or you want to look at an area that's expanded. So we have a method where you can essentially do a matrix multiplication to look at the relationship between the tract volume and the surface of the particular brain, and you end up with maps that look something like this. And so we've done this for several species, and we feel that it passes, a basic test of it does represent where we expected some of these major tracts to go. So this is a human example where our arcuate fasciculus hit the areas that we wanted to hit. We have some lower probability hits on some areas that are, a little less likely. But that said, it was a large population, and we know the arcuate is quite variable. Our SLFs are quite contained where we expected them to be in terms of what we know about the anatomy of these dorsal tracts that connect parietal areas to frontal and prefrontal areas. So what we have been working on then is doing this across species so we can directly compare not just tracts, but then tract surface projections across species. And I wanted to zoom out here just for a moment to talk about what kinds of questions can you look at with this kind of data. So one of the things to think about when you're thinking about brain evolution is, so we know brains can get larger if there's selection pressure for larger brains, and we know they might add additional sulci, but there's all kinds of other things that can happen as brains get larger. So we can have something like beyond a shared ancestral state, we can have just an increase in brain size, and that certainly can happen. We could even call that the null hypothesis. Perhaps as brains get larger, the organization stays the same. It's just a larger brain. We can have selective expansion. So we can have, like I showed you earlier with the middle temporal gyrus, I would argue is a bit of a selective expansion for a lateral temporal cortex. And then you can start to ask questions about what selective pressure would have caused a regionally specific expansion. We can have what's called increased arealization. So what that means is you have an area, maybe it's already been well characterized in a macaque, for example, and then you look at a human or another primate and you see, oh, it's actually connecting up in different ways, or it seems to be subdivided. Now this area is doing multiple things and it's kind of breaking into different components. And we can have, on a connectivity level, all kinds of things can happen as well. So not just, oh, there's a new projection of this tract, but also maybe the strength of maybe the tract volume has gotten larger, the strength of the connectivity of the tract has changed. So because I focus on white matter tracts, but I just do want to point out, you can do this with all kinds of other data as well, if you wanted to do evolutionary approaches to brain anatomy. So like spatially organized gene expression profiles, like spatial transcriptomics would be a great one. Myelin, of course, all kinds of things. So, and then you can do these interspecies comparisons. So essentially, this is all moving towards, we want a common space where we have features across the brain that we can then directly compare. And the nice thing is, then once you have this idea of this common space, so a brain by feature matrix, not only can you compare species, but you can also compare individuals across development. You can, you could compare like a neonate human to a neonate other, like a marmoset, for example. You can do all kinds of comparisons. You could do patient groups as well. So I'll talk a little bit about what kinds of human temporal lobe specialization. I'm going a little long, aren't I? Okay. So I'll tell you a little bit about some of the data using this approach we found. So once we do the direct comparison, we can end up with a sort of heat map. And basically, we can start to see what are the areas that are the most different between two different species. So the hottest areas are areas that when in this particular instance, we compared a human to a chimp. These are areas in the human that have a connectivity profile based on all those tracks we reconstructed that is the most different from the chimp. And the areas that are dark, that says, you know what, this had a connectivity profile that was very easy to find in the chimp, matched quite well, right? So in this area, for example, say, well, there's really no difference. It looks like humans and chimps have very similar organization in terms of long-distance connectivity. But we found these big hotspots here in the temporal lobe and in the parietal lobe, which was very exciting. And I'm going to show you just how the process works. So once we know what the hotspot is, we can then interrogate the vertices on the hotspot and say what tracks are driving these differences. And in this case, we found arcuate fasciculus in the left hemisphere, which we were happy about because we know that is important for human brain specialization. But we also found differences in this other fasciculus called the ILF. And so going back to my earlier points about primate temporal lobe, the ILF is a tract that connects visual areas and it runs along the length of the temporal lobe. We also have data from other papers suggesting it is expanded in both humans and great apes. So we seem to have an extra segment. So this is where I'm very interested in the temporal lobe in terms of a locus of not just primate evolution, but perhaps humans and great apes have also had some interesting new specializations around the inferior longitudinal fasciculus that we don't find in other primates, and that has implications on how humans process visual information. So just quickly, I'll briefly mention. So this is in review right now. We've kind of interrogated more carefully what's going on in these different areas. This was bilateral, this AF, this arcuate fasciculus and ILF finding in the posterior temporal lobe. It's very, very large. So that tells us that between humans and chimps, humans have just had a lot of reorganization of posterior temporal lobe. We also found this inferior parietal lobule difference that was driven also by this ILF, this change in this ventral, what I might say is part of the ventral visual pathway. So that also suggests some really interesting differential organization. I also want to point out what was neat was, maybe will make some people irritated, we found very little prefrontal or frontal differences, which I think people expected. But when we looked at the differences when the macaques, they popped out. So don't worry, your prefrontal cortex is different from a macaque, but it might not be different from a chimpanzee. I'm really sorry. So just to wrap up, we can sort of combine this information with functional information. So, many people have done many fMRI studies, and now we can use databases to query if we segment the areas that we found were high, what are they doing? And it's kind of what we expected. Language, also the right hemisphere bias of our IPL finding is interesting because it implicates TPJ, which is a social cognition hub, arguably, in humans. So we found that very interesting, and it suggests that we can perhaps bridge the gap between some of these anatomical observations and what the implications there might be functionally. And I think I'll stop there. Thank you very much.
Thanks a lot. We're going to have a few minutes for questions. I have one question. I wanted to ask you about the arcuate fasciculus and the 3 segments of the arcuate fasciculus. So since you have those differences with the parietal lobe, do you think that this is related to different segments of the arcuate?
Yeah, and actually I didn't have time to let me. Would it be okay to have my slides up again? Okay, so yeah. Fortunately, I ran out of time. But that's when, with Joanna Sierpowska, we worked on this question because, she understood the anatomy of the arcuate very well because of her clinical experience. And so we were the first to look at the different segments of the arcuate fasciculus in chimpanzees as well, and we were able to reconstruct them. And then we were able to identify how the changes of the organization of the arcuate fasciculus. And essentially what we found was it seems to be more connected to prefrontal areas than we expected. And we basically looked at 2 major language hubs, and we found essentially the frontotemporal component of the arcuate has featured a lot of changes in humans specifically, but some of the other components of the arcuate are more conserved. Okay,
thanks. Any other questions?
Hey, thank you for your talk. I had a question about the endocast. So you mentioned at the very beginning of your talk that you could do endocast on fossils. Can you really see the sulcus when you do the endocast, and then can you compare the position of the different sulcus on the endocast of fossil primates?
Yes, it's not perfect. So as you might expect, it's a brain-like impression. So when the animal decays, it leaves some imprint onto the cortex, but they're not going to be deep. So we're not going to be able to go deeply into the sulcus, but we can get essentially clues about the surface sulcal information. And actually, some of these images here are because I have not been able to find anyone who has, for example, a picture of a eye brain, but I have a colleague in Belgium who has tons of endocasts. And so comparing those endocasts, like here's an injury to this lemur live brain, I feel like it looks very similar. You have a lot of the similar information. Yeah, I would say, yeah, you can't measure the inside of the sulcus, but yeah.
Okay, last question.
Thank you very much. Great talk. I was struck by how blue the prefrontal cortex is, suggesting that there is not massive changes between species in here. And I was wondering whether this could be due to the method that you use. So if I understand correctly, yes, you do this comparison based on the idea that there exist similar tracks between species. But yes, if you have an area which expanded massively, then you cannot have this comparison in the first place, and you may not get these results.
100% agree, and I should have made that more. So this is with our approach, looking at it could be, for example. So there's many papers showing that especially the prefrontal cortex in humans has selectively expanded more so than the temporal lobe even. So let me see this one. So we may be seeing a change in overall size, but the connectivity related to the major tracks has not changed, and that's still an important change. And another paper I didn't have time to talk about is a recent paper showing that the pattern of gray matter loss in aging in humans, it seems to correlate quite well with areas that have expanded the most, and so that's why it may be why we see so much prefrontal loss in humans, because it's an evolutionary recent expansion. So yeah, I think if we did this approach to brain and feature matrix with something besides fasciculi, we would get a different answer. Yeah.
There's a last question from someone online.
Okay.
So thank you for this fascinating presentation. I'm particularly interested in the potential of virtual dissections. Is it possible to use this technology to explore the brain in greater depth, especially focusing on subcortical nuclei?
Yes. So I'm actually able to reconstruct tracts that are coming from the thalamus and some of the smaller tracts. But as we are getting better and better, higher resolution scans, that's something that people are starting to look at. So I'm looking at, and I should mention, I can track from the LGN and the MGN, depending on what this person's interested in. But I think that's the future, is then getting down into these smaller subcortical nuclei and seeing their relationship to cortex. Absolutely.
Thank you very much.
Thank you.
So our next speaker is Marta Matamala from the Cognition and Brain Plasticity Unit, and she will show us her research about sensorimotor cortex synchronization with auditory and visual inputs related to walking movements. Thank you very much, Marta. We are looking forward to your presentation.
Okay. So thank you so much, Ruth. And also thank you so much to the Scientific Committee and the Institute of Neuroscience for inviting me to give this talk. It's an honor to talk after you guys. So I really enjoyed your talks. And well, so today I'm gonna introduce some of the first studies that we are developing in the lab in the frame of my Marie Curie research project, that is about investigating the impact of auditory inputs to improve the walking ability in patients with a stroke. But first, in order to know how auditory inputs can help to enhance this walking movement in patients, we decided to investigate the impact at the neurophysiological level. So we conducted two studies to investigate this. This is the first one study that I conducted with Andrea and other colleagues. Now this study is under review. And so in this study, we wanted to investigate how auditory inputs related to the walking movement, that it's using footstep sounds, can enhance the synchronization with the sensorimotor cortex compared to visual inputs in a walking movement integration task. So as Andrea already explained, so his talk was really helpful for me. So there is a huge amount of evidence showing how auditory inputs can really synchronize with the sensorimotor cortex when presenting them at specific frequency rates. Especially, evidence has shown that when the auditory inputs are presented at a frequency range of 2 Hertz approximately, there is a proper synchronization with the sensorimotor cortex, as we observed in the video of the parrot, for example, how the movement of the parrot was synchronizing with the auditory inputs, that he was listening. And so about the auditory inputs, we had a lot of evidence, but also related to visual inputs, and related to visual inputs showing body movements. There is some recent investigations, as this one from Krakow and colleagues, that is it's a paper from 2021, in which they shown how the brain can also synchronize with visual inputs presented at a specific frequency range that also is around 2 Hertz. So in this paper, the authors use a point light figure presenting a walking movement, displaying the movement at a frequency of 2.4 Hertz. And the authors observed that the brain oscillatory activity synchronized also with the visual inputs at that frequency range in the occipital cortex. So, as we said, or as I said and Andrea said, so the neural entrainment is the synchronization of the brain oscillatory activity with external sensory inputs arriving to our nervous system. It's this ability of the neural system to synchronize with sensory inputs. And one specific approach that is a recent approach that is now being used is the frequency tagging approach, in which consists in displaying the sensory inputs at a specific frequency rates to investigate if the brain is able to induce this effect of neural entrainment with the sensory inputs. So in our investigations, in the 2 studies I'm going to present, we use this kind of technique with an EEG, with EEG recordings, the frequency tagging approach. So let's start with the background that give us to conduct this study. So related to the auditory inputs, related to body movements, the investigation to investigate how auditory inputs can be useful to investigate the synchronization of brain installations with body movements. There are some research arguing that when you couple auditory and visual inputs related to the body movement and specific to the walking body movement, you can get a higher synchronization with the brain oscillatory activity. But other studies says that the visual inputs are the crucial inputs to induce this brain synchronization. But there are other studies showing that the visual cues are not relevant to induce this brain synchronization of the walking movement in the brain, no? So in this study, we wanted to compare auditory input related to the walking movement that they were like footstep sounds, and also visual inputs related to the walking movement that was the walking movement displayed with a point light figure. So the main aim of this study is to investigate rhythmic sensorimotor synchronization when coupling auditory and visual input related to walking movement using the frequency tagging approach. So in this first study, we used 22 healthy subjects without presenting any visual or hearing impairments, and we did the recordings with a 64-channel cap. This is the experimental paradigm in which we had 3 different experimental blocks. The first one was the visual block, in which the healthy subjects, the subjects could observe a visual input that was a point light figure walking in rhythmic and random sequences. Then we are going to see a video and it will be clear. And the rhythmic and random sequences were presented at different frequency rates. A frequency rate of 1, 2, and 3.6 hertz. And each frequency, each condition in rhythmic and random sequences were presented 4 times. And so this was a first block of the experiment. Then we have the auditory block, in which the participants could hear like footstep sounds displayed also in rhythmic and random sequences, and also at 1, 2, and 3.6 hertz. And then there was another task that was the audiovisual task, in which we coupled the auditory and visual inputs also at rhythmic and random sequences, and also presented at the different frequency rates. So between the different experimental blocks, there were a period of pause, and the whole experimental procedure lasted 3 hours approximately. And so in the auditory task, you can hear the rhythmic footstep sounds. Can you hear? Yeah. This is the rhythmic footstep sounds presented at 2 hertz. We presented also at 1 and 3.6, but this is just an example. And in the random footstep sounds, I think that it was no. This is the random sequences. You see, the random sequences were sequences without following any rhythmical pattern. And also in this task, in the auditory block task, the participants had to conduct an attentional task to ensure that they were paying attention to the stimuli. That was to identify different changes in the pitch sound of the footstep sounds. There was a variation of plus and less 50 decibels, and they had to report us at the end of the presentation of the sensory inputs how many changes they could identify. In the visual experimental block, we had the rhythmic sequences of the point light figure walking. This is at 2 hertz too, but there were like 1 and 3.6. This is the rhythmical one. And let's jump to the random one, is this one, in which you see there are like random movements of the body, but the body movements are not following any rhythmical pattern. So in this task, the participants had to conduct also an attentional task that identifying changes in the red dot here in the middle of the body of the point light figure that sometimes changed to white color. And at the end of the sensory input presentation, they had to report us how many changes they identified. And in the audiovisual task, the participants could hear and observe the coupling of the auditory and visual inputs presented at the different frequency rates. And this is the random sequences. Okay. So in this task, they had also to identify the changes in the red point in the middle of the body of the point light figure. And each sensory input lasted 60 seconds. And during this period, we were recording the oscillatory activity of the participants. So for the pre-processing and processing of the data, we used for the pre-processing the EEG lab toolbox, and we applied the ICA, the independent component analysis, to remove the blinking and the muscle noise of the source of the signal. And then we use also the current source density transform was applied to the whole data, and we epochted the data, the 60 seconds signal in epochs of 20 seconds for each sequence, and each sequence was also baseline corrected using the mean power activity of the whole sequence. So we took the spectral power and using the field tape toolbox in MATLAB, applying the multi-taper method, and also basically applying a fast Fourier transform and using the hanging window. We analyzed the data of the 22 participants. Then we analyzed the mean power activity with a general linear mixed effects model. And the model includes the rhythm condition and 3 different regions of interest that we designed to investigate the power activity. So these were the main regions of interest that we found that are important to our experiment. So we designed these three regions of interest: the sensorimotor cortex, the temporal cortex, and the occipital cortex, to see how they synchronize between them when displaying the sensory inputs to the participants. So in the results, you can observe in the left side the rhythmic sequences, the results in the rhythmic sequences in the waveforms, and at the right side the results from the random sequences. And as you can observe in the rhythmic sequences in auditory, this get crazy. In the auditory task, there is a peak at 2 Hz in the sensorimotor cortex and also in the temporal cortex. And also we found this result in the audiovisual block, in which we observed a huge peak at 2 Hz and in the sensorimotor and temporal cortex, but we didn't observe this peak in the visual task. We observed the peak at 2 Hz, but only in the occipital cortex, so not in the sensorimotor cortex for the visual inputs, only with the auditory and audiovisual blocks. In the random sequences, as you can observe, there is no evidence of any peak of activation. And if we took the difference between rhythmic and random sequences, we observe also a huge peak in the auditory, for the auditory task, in the sensorimotor and temporal cortex. So apparently the temporal cortex synchronizes with the sensorimotor cortex when displaying the auditory inputs. And also we observe a peak at 2 Hz in the sensorimotor, temporal, and also in the occipital cortex in this task. And again, in the visual block, we couldn't observe any peak at 2 Hz. We observed this peak at 3.6 Hz, but so based in the bibliography, this is more related to the processing of the visual input. It's not related to rhythm, especially. And in the topographies, we observe also same results. We observe that at 2 Hz, we obtain a greater synchronization of the sensorimotor cortex in the auditory task, as you can observe at 2 Hz. And also in the audiovisual in the audiovisual task, we observe a high synchronization with the sensorimotor cortex at 2 Hz. And yeah, we could, and one in the sensorimotor cortex, but also in the occipital and temporal in the audiovisual condition. So, as a first conclusions of this study, we observed that rhythmical sequences can induce a greater brain synchronization effect in the brain selected areas, and especially the auditory inputs using the footstep sounds presented at 2 Hz induces a synchronization, a greater synchronization with the sensorimotor cortex compared to the other sensory inputs. And the synchronization effect with the sensorimotor cortex apparently was enhanced when coupling visual, auditory and visual inputs presented at 2 Hz. And this is one hypothesis. So we hypothesize that our results are with the auditory inputs can be mediated by a greater attention toward the auditory inputs compared to the visual inputs. So now, very fast, I have time or not? No. Okay, so nothing. We observe same effects in patients with a stroke at 2 Hz. So super fast. It's like, you can observe here in the topographies, we compared healthy with patients with a stroke. And we wanted to observe if this could be applied to patients with a stroke. And we observed that they have the ability also, even if they have cortical impairments, to integrate these auditory inputs and induce the synchronizations with the sensorimotor cortex. Well, and nothing else. And this could be helpful for future rehabilitation trainings using auditory rhythmic stimulation in patients with a stroke. So thank you so much to all my research group and my supervisors of the Marie Curie project.
Thank you very much. Yeah, so we have a few minutes for questions. I wanted to ask you about the results of the stroke patients that you commented very, very fast. So in the stroke patients, which kind of stroke did they have?
It's like all of them were ischemic stroke patients, and they presented, some of them presented a cortical and some of them subcortical injuries. And there were chronic strokes, patients with chronic stroke.
Okay, but the lesions were in areas that you would expect to affect rhythmic processing or sensorimotor synchronization?
Well, if they present injuries in the subcortical structures, probably the processing of the rhythmical inputs can be impaired because of the injury. But we observe that even if we are still working on the analysis of this data. The overall results show that they have the ability to process the rhythmical inputs, as in healthy patients, but we have to still work on the correlate, so split the data in patients with cortical and subcortical injuries. So we still don't know very sure if the subcortical injury has an affection in the processing of the rhythm, but the overall view of the results shows that apparently there is no an affection. So they could integrate well the rhythmical sensory input. Yeah.
Any other question? Okay, thank you.
Thank you for the presentation. How do you expect to translate the results of those experiments to the rehabilitation of the patients?
Well, is that there are like some rehabilitation trainings that use auditory input stimulation with metronome to improve the walking patterns of the participants. And well, the idea should be that to use the auditory inputs like maybe footstep sounds displayed at this specific frequency range of 2 Hz, that it will be around 120 BPM. It's a bit fast, but apparently it's the frequency that works better to integrate the movement, the walking movement. So you can couple this and ask the patient to try to follow the rhythm of the you are displaying them. So there are some therapies already on that, but not using a specific rhythmic patterns and not fixed rhythmic patterns. So they use music and other like type of rhythmical stimulation, so music, auditory stimulation with specific beats, but this with a specific frequency range is not done yet. Yeah, thank you.
Okay, so we will now move on to the next speaker. Thank you very much, Marta. So our next speaker is Marina López-Solà. She's also from the University of Barcelona, School of Medicine, the Institute of Neuroscience, and she will explain why we need brain biomarkers of pain. Thank you.
Thank you so much, and it's really an honor to be here presenting today after such great speakers. I'm going to switch gears a little bit and talk about pain. And since we have very little time, I'm going to try and be a bit expedited. I'm going to try to show you that psychologically informed brain markers of pain are actually relevant and necessary. The first situation that I will try to show you with my presentation is that symptoms of pain are actually not sufficiently a good guide to treatment selection. We really do need to understand what are the pathophysiological mechanisms that actually can account for the symptoms of pain, and we need to have measures of that if we want to adjust treatment appropriately. The second thing is that actually the psychosocial context and the meaning that we attribute to both pain and treatment are actually important in the process of developing brain biomarkers of pain. So this thing, pain, is actually a root problem, and it's a root problem not only in adults. It really starts in adolescence and plagues adolescents. We have 20 to 35% of the population in chronic pain. This has been rising in the past decade if we take into account recent epidemiological studies. The costs are huge, around 600 billion euros annually. This is only in European countries. And as I was saying, chronic pain is already very frequent in adolescents. We have up to 37% of European population in chronic pain during adolescence. This is particularly relevant because more than 2/3 of these kids actually persists into becoming adults with chronic pain and do not respond to current treatments. This type of problem has very long-term negative consequences, particularly if we talk about developing youth. It has been shown to compromise physical activity, social relationships, mental health, academic performance, and directly future professional development. But what are the mechanisms, and how can we predict if somebody is at risk of having a persistent trajectory? Up to now, we have to think that this is actually the gold standard and the only information that we use many times to adjust pain treatment in the clinic, this type of scales. This tells us about the subjective experience, although in a very limiting way, but it doesn't tell us anything about what's driving the symptoms. So let's have a case study here. We have a 65-year-old male. So that's a person that is actually, in principle, very protected for pain, also highly educated, high socioeconomic status. This person has a back strain while loading a suitcase in an airport, and 2 weeks later develops lower back pain with a spasm, then left lumbar area pain. Then, after thorough examination, they think this person has a disc herniation. But when they look at the MRIs, they see no signs that could account for the pain. During 5 months, this person is twice daily in physical therapy, acupuncture, deep tissue massage, postural work, no pain relief whatsoever. At 7 months, pain starts migrating to other regions that are close to the main region of pain. He starts losing the strength in the leg. The doctors suspect nerve entrapment, but actually injections of steroids and anesthetics are 100% ineffective. This is actually the person I'm talking about. He wrote about this. He used to be a dean at Stanford University School of Medicine, a person that was very powerful, very influential, that was very carefully looked into, and they could do nothing for his pain. He says, as time went on, my symptoms and limitations worsened. Various clinicians offered suggestions. Some wondered whether the pain was becoming, in between quotes, functional. Others strongly believed it was probably neuropathic in origin, but without a defined trigger. Progressively, he entered a trajectory of depression. This person was strongly adaptive. He had been running marathons. He had been one of these hyper performers. And he ends up saying, I had sometimes wondered whether the chronic pain that patients reported was as incapacitating as they claimed. I now know it can become debilitating, sap one's energy, negate or neutralize joy and well-being. And I'm glad he wrote about this, because with his voice, he actually made, at least in that territory, an impact. Here I'm showing you the journals of our teens. This is the Sprint study, signatures for trajectory of chronic pain in adolescents. This is the first study done in youth with chronic pain. Here we have the case of an adolescent, one of our adolescents that actually did not get better at all. This person started receiving multiple surgeries and medical procedures to address the brain problem and frustration. And you see from 2016 to back here in 2022, their last sentences are, why can nobody help us? It never lets up. This person, 1 year later, is in very high pain trajectory, deeply inhabilitated. Another case of high persistent pain, as per the diaries here, when I think about my pain, it's going through a straight line. Hills and throws, it often proceeds in spikes. Another highly prevalent, besides having undergone all sorts of currently available therapies, combined the way it's been possible. Here we have another case of a person that, I would see all the kids playing and I would feel super jealous and sad. Then a number of things happened, many, again, different treatments, but in this case, the person, food got better, could play soccer and run again. This is a successful trajectory after treatment. So basically, we don't feel we have any way to predicting who and how are going to get benefits from these treatments. We have been throwing darts at the wall and seeing what sticks. That's exactly the National Director of Policy and Advocacy for the US Pain Foundation. So he's not nobody with regards to this problem, right? What if we were better at hitting the target? The focus up to now has been on the body. For example, but look at the problem of the body, okay? Imagine findings in the back for people with no back pain. Check it out. Disc herniation, age 40, 68% of people that have no back pain actually have disc degeneration upper imaging. Disc signal loss, 54%, and these numbers do only get worse as we age. These changes are typically like wrinkles, forms of aging, okay? Here we see another example in the natural reabsorption of herniated discs 1 year later. So this cannot be the issue that solely maintains the pain, okay? Here we have another case in osteoarthritis of the knee. Pretty much they conclude the correlation between tissue findings and pain seems weak at best. Hence, the problem seems not to be in the body. It does not account for the symptoms the patients have. What are the pathophysiological mechanisms? And here we come with the idea, well known, everybody, many people are working on this. What is a biomarker? So it can be defined as a symptom-linked biological measure that has high sensitivity and specificity to identify pathology in new people, okay? We can derive them from blood, from body responses, from saliva, and here we are going to be centered in the brain. We have different types of markers that could be relevant, related to diagnosis, to prognosis, trajectory, to stratification of patients, and to identifying particularly important psychological constructs that we know have a big influence on pain. For example, brain markers of acute nociceptive pain, brain markers of fear of pain, brain markers of fear of movement, of negative affect, of negative self-referential functioning, of multisensory unpleasantness, all of these constructs that are severely associated with chronic pain. So The idea is we are trying to put a lens into what's going on so that when we go with treatment, we go to actually hit the pathophysiological mechanism that is being expressed in that particular person. Why brain biomarkers? Because there is evidence of neuroplasticity in animal models of chronic pain. We started in the animals. There we can control the injury, and what we see after the injury is resolved is persistent pain behavior, neuroplasticity, big neuroplasticity, systemic inflammation, and neuroinflammation. There are different mechanistic components. Some of the plastic changes represent the immediate experience of pain. Some others are really related to sensorimotor aspects of the problem. Other issues in the brain are related to valuation, affect, and associative learning associated with pain. What we need to know, to be kind of inspired in animal models so that we can go to the humans and check what's going on there, which mechanisms matter there, and what is the locus of pathology in a given subject. This is essential to be able to match the treatment. Most of the research up to now has been done in adults. Here is the complex picture. We know that pain is an emergent property from coordinated brain activity that span multiple regions and systems. So basically the input goes up into the thalamus and also the parabrachial nucleus. Those are direct anatomical connections, and expands along the cortex, S1, S2, posterior and anterior insula, anterior cingulate cortex, and subcortically the amygdala. All those directly receive projections that are pain-specific. So pain is actually a highly distributed parallel process. It's not serial, it's not in any way composed of one mechanistic aspect. So basically, we think that there are going to be different brain biomarkers of chronic pain, and that chronic pain will be best explained with multiple component markers. For capturing that complexity, multivariate approaches spanning the whole brain are going to be the ones that are going to give us better and more robust information. We are trying, therefore, to capture spatially distributed patterns of brain activity or brain connectivity that span multiple brain networks. Here, I'm presenting you what was the first brain marker for nociceptive pain. Here we have the anterior cingulate cortex, weights in the posterior insula, S2, etc. You see that there are profiles of heights and valleys of brain activity during nociceptive input that best predict how much pain you have. This brain marker, you see, is predicting pain very, very strongly. It has accuracy in new subjects above and beyond 94%. This has now been replicated in 60 samples all over the world. And we know that this brain marker of pain, of acute nociceptive pain, is actually sensitive to gastric distension, esophageal pain, rectal pain, vaginal pain, noxious pressure, electric shock, and noxious heat. And it is not sensitive to constructs that are psychologically very close but distinct, such as aversive images, social rejection, observed pain in strangers, pain anticipation, pain recall, etc. So right now we have one component marker, okay? One. So, okay, this one we know is associated to the quantity of nociceptive specific signal that the brain is processing, okay? Okay, does this brain marker capture all forms of hyperalgesia or analgesia? Check it out. Here, this was a simple experiment in which what was demanded to the participants was to either regulate pain up, oh, you are not, I'm sorry, are you seeing me? If I do this, you see the arrow? Okay, so here you see, those are stimulus intensity, pain rating. Here you see the pain rating goes up very strongly with stimulus intensity, as one way may think. This effect of temperature is very strongly associated with MPS response, neurologic pain signature response, that marker. But check it out. Sometimes they ask the subjects, hey, regulate your pain up. Think that your pain is really, really nasty, that it's going to put you into a bad trajectory, that, okay, they gave them a specific clues to regulate pain up. They indeed managed for pain to go up. They were feeling more pain under this condition. And the opposite was happening in the regulate down, blue condition. Check it out. These effects of regulating up, cognitive reappraisal, regulating down, cognitive reappraisal again, have zero effects on the MPS. They do not touch nociceptive pain-specific signal in the brain, but they are analgesic. This means that the analgesia is happening through a different mechanism. So indeed, the neurologic pain signature does not track all forms of pain. Here we have a summary of all studies done today on placebo analgesia. Placebo analgesia has a huge effect on reducing pain. Hedges G of minus 0.66, strong effect, happening in 17 out of 20 reviewed studies. Yet check it out. Effects on the MPS, on this neurologic pain signature, are virtually zero. So definitely placebo analgesia is not acting by reducing nociceptive specific signal to the brain. Now let's check it out. This is a study that I did with osteoarthritis patients. Here, what we were looking at is, okay, does naproxen, which is a strong anti-inflammatory drug, at a single dose, reduce the amount of pain that these patients feel in the right spot where it's most painful of the knee? And what we found is that, and then does the MPS track this analgesia? We found, okay, what we found is like no effect whatsoever of placebo, neither on, here there was effect on pain ratings, but not on the MPS, zero effect on the MPS, and a moderate, small to moderate reduction of the MPS, clearly, beyond placebo. Just one dose of naproxen. This effect was comparable to the reduction in pain ratings that we observed. So in this particular case, naproxen indeed reduced nociceptive specific signal to the brain. We don't know if it may have reduced other mechanisms. We know it reduced one. But check out how beautiful this is. This is, we took female women, more or less my age, a bit younger, in the scanner. And they had a partner. And we put sometimes the woman to feel pain only holding a rubber band. And sometimes, how am I doing? Very bad. 1 minute. Okay. And sometimes they were holding the hand of their romantic partner. So what we found is that just holding the hand of the romantic partner during pain had a profound analgesic effect that was very strong in pain and pleasantness, and it increased emotional comfort very significantly. And what we found is that this specifically reduced the MPS. Holding hands with a romantic partner reduced the MPS a little bit more than naproxen did in OA osteoarthritis patients. Here we see mindfulness meditation, attending moment by moment to the sensations in your body while you experience pain with neutrality, not trying to hold them off. Check it out. Neurological pain signature, control group, placebo, no effect whatsoever. Sham mindfulness, no effect whatsoever. Check it out. Mindfulness meditation, very strong effect. In two completely independent studies, there is a higher drop in this study. In this other more modest study, 26% drop in the neurologic pain signature. Strong effect, okay? And here I wanted to tell you, hey, what does the MPS tell you about a disease such as fibromyalgia? You know fibromyalgia, chronic widespread in the 4 quadrants of the body, and this is the only thing I will tell. Here what we saw is, check it out. Here we have control women stimulated at 4.5 kilograms force, control women at 6 kilogram force. You see there is a huge increase in the expression of the MPS. Okay? Very strong effect. Now let's see. How do fibromyalgia women, which are hypersensitive to pain, respond to 4.5 kilograms force? Exactly the same, exactly comparable to 1.5 kilograms force more in the control group. This is directly talking about augmented bottom-up nociceptive specific signal in the brain. Is this all the story that there is? This explained, by the way, acute hyperalgesia in patients. Is this the whole story that we have about fibromyalgia? Not at all. I could tell you a little bit more about how multisensory processes, specifically in the brain, are deeply altered and how do they contribute to fibromyalgia. Unfortunately, I don't have time. I had a couple more studies, but yeah, later we can talk about it. I appreciate it very much. Thank you very much. Thanks.
Thank you very much. So we have a few minutes for questions.
So thank you for your talk. Regarding the fact of the mindfulness that reduces pain a lot, do you think that because they are like paying attention to their own body because of the mindfulness technique, but it's a guided meditation. Okay, so it could be that
in one of the studies. In the other one, actually, they had been trained before, and they underwent the meditating state on their own.
Okay. And do you think that it could be like also for mechanisms of distraction of pain? So, I mean, that they are not focusing the attention on the perception of pain, but it's more like a distraction of the painful.
Definitely, it could be the case of distraction, but it seems to be a little bit more nuanced. And the same holds true for the hand-holding component, because we checked distraction-related networks, and it was not mediated by that. In the case of mindfulness, actually, when you go and you check out where the expression of the marker goes down, okay, it's very interesting because it really does not go down in all places the same way. So what we've noticed is it really goes down much more on anterior cingulate cortex, which would be more like and all the anterior component at the ventral level. And interestingly enough, because they are actually paying attention to their body, as you very well said, there seems to be a breakage of the network such that there is augmented responses in S1, S2. Mm-hmm. And there is diminished responses in the more affective, which is actually the most anterior and also ventral part.
So you think it's mediated by affective?
It seems to be breaking up the normal process, enhancing the fine quality of sensory processing, and reducing the response level.
Okay,
that's what I think.
Thank you. Thank you so much.
Any other question?
This is really interesting and important work. To what extent do you think that in the near, hopefully, future, we will be able to use modulatory neurofeedback techniques in order to teach patients to self-modulate the activity of this network, and to what extent we have already or not demonstrated that downregulating it will result in feeling less pain? Mm-hmm. Thank you.
Thank you very much for the question. Yeah, actually, this is already being done by a colleague. His name is Philip Craigle. I think he's in Atlanta now. He was in Emory University, then in Colorado. I think he's in Atlanta. And he is already working with right now we have this MPS that I talked about, but we have markers of negative affect that are very specific of negative affect. And he's working to try to see if regulating one or the other marker gives you better outcomes at analgesic behavior, at analgesic patterns. For what I know, I don't know if he has already published this, but he was obtaining pretty cool results. And he was trying to tailor it. What is most helpful? Is it most helpful to concentrate on the expression of the marker on S1 and S2 on the posterior insula? Or is it more helpful to really take the whole marker, or to take the marker in anterior regions of the brain? So those are I know they are doing that, but I'm sorry, I cannot give you the full answer.
I have another question. Yeah, I wanted to ask you about the developmental part. So you've mentioned some work with children. There are huge individual differences in children, how they respond very early on to pain. Do you think there's also something there in how they cope with pain for then the future?
Yeah, there is a really big. We are working with this study. It's the SPRINT study that I didn't get a chance to present with you to you. And what we are seeing is these huge individual differences that you are talking about, and they seem to be at multiple levels, not only at basic quantitative sensory testing metrics, which already show huge individual differences, but also at coping strategies and at cognitive flexibility and at what is called resilience from a cognitive psychological standpoint. We see approximately 1/3 that is very highly resilient, and they may have really strong pain, but they figure out a way to perseverate on their aims and to be very flexibly adapting what their strategy should be as a function of their life aims, their purpose. Whereas there are approximately 2/3 of these kids that do not have that. We started working in a separate study, a study that I led in the University of Cincinnati, And we were studying juvenile fibromyalgia patients, girls from 13 to 18 years of age. And what we've seen is that these effects are mostly mediated by the way that the dorsal aspect of the default mode network flexibly connects to other networks. That is very helpful in which of them are going to show the executive function abilities and this kind of switching attention in a most beneficial way. And that, again, in that case, we separated from the most severe patients 2 big groups. 50% were high resiliency, 50% low. And we saw that result that was coming up very strongly in the dorsal default mode network that, you know, is related to many things, but also to cognitive flexible attentional focusing and to cognitive flexibility, amongst many other things related to self-referential thinking. Self-referential thinking and the importance that pain has in self-concept, which is being studied by one of my grad students right now, is another core element. And that's where the most variability we have amongst, right?
Okay. Thank you very much. Thanks a lot to everybody.